Hi everyone, my name is Duaa. Today I'm going to present my project on evaluation of gene gun technique for transient expression of the Q8V3T9 viral fusion glycoprotein in plants. Bialistic transfection is a well-established method for delivering DNA into a wide range of plant cells. It involved the physical introduction of DNA-coated particles, typically gold or tungsten, into plant tissue using high-pressure systems. Recent studies, such as the tool to shoot gene guns with a man-made air pressure, have made this technique more accessible by providing a cost-effective and portable alternative using a simple air duster gun and a bike pump. It offers a practical approach suitable for both research and educational environments. Transformation efficiency is influenced by various factors, including delivery pressure and plant tissue characteristics. This study aimed to evaluate a low-cost, in-house gene gun system for transient expression of the Q8V3T9 viral fusion glycoprotein tagged with the red fluorescence protein M cherry for visualization in various plant species. The effectiveness of the construct was tested on onion, bok choy, and lettuce leaves, with the hypothesis that the gene gun would facilitate successful transient expression under optimized delivery conditions. The experiment began by streaking E. coli containing a TI plasmid onto nutrient agar containing kanamycin. After overnight incubation, a single colony was inoculated into nutrient broth and cultured. Plasmid were then extracted using the gene elute mini preparation kit and alkaline lysis method. The in-house gene gun was assembled using bike pump and air duster gun. Bullets were prepared in two types, with the plasmid and without a plasmid as a negative control. DNA-coated gold particles were prepared and delivered into leaf samples at two different pressures, 40 psi and 100 psi. Samples were incubated for 24 and 48 hours. Transfection success was evaluated using fluorescence microscopy with Texas red detection, and the number of fluorescent cells was quantified. To confirm transformation, plasmid recovery was performed from leaf tissue and analyzed via agarose gel electrophoresis. The results showed that onion samples transfected at 120 psi and incubated for 48 hours had the highest number of fluorescent cells, followed closely by bok choy under the same condition. Lettuce showed the lowest fluorescence and higher background, making it harder to interpret. However, high pressure also caused visible tissue damage, especially in lettuce, which may have led to false positive in control samples. This suggests that leaf thickness plays a role in how well tissue tolerate delivery force. Thicker leaves, like onion and bok choy, handle the pressure better. When comparing the two pressure settings, 40 and 120, used of onion leaf sample, 120 psi resulted in higher number of fluorescent cells, with a significant difference between samples treated with plasmid and those without. Fluorescence imaging clearly showed red signal in successfully transfected samples, while the negative controls also exhibited some signal due to leaf damage. DNA extraction was performed only on onion leaf samples to confirm successful transformation. The extracted samples were run on an agarose gel. The results supported plasmid recovery, as faint bands were observed in two of the samples at the expected size, while the third sample did not show a band at that location. The bands near the bottom of the gel represent the genomic DNA for the onion leaves. In conclusion, this study shows the successful use of cost-effective in-house gene gun for bialistic transfection in various plant species. Among the sample tested, onion leaves had the highest transfection levels. The best results were seen at 120 psi after 48 hours of the incubation. Higher pressure did improve transfection efficiency, but also caused visible tissue damage in some cases. This highlights the need to balance delivery force with tissue tolerance. Fluorescence imaging confirmed the expression of the M cherry tagged transgene. Plasmid recovery and gel electrophoresis provided further verification. These results demonstrate that affordable biolistic system can be effective for gene delivery. They offer a practical option for both research and educational use. To improve and validate the biolistic transfection protocol, several future steps are planned. Testing higher pressure setting may boost transfection efficiency, especially in thicker or tougher leaf tissues. The study will also expand to include more plant species and leaf types. This will help assess how versatile in-house gene gun is. The plasmid recovery process will be refined to increase yield and make transformation confirmation more reliable. In addition, PCR with specific primers will be used as more sensitive method to detect transgene. At the end, I would like to thank Dr. Jessica Livingston, Dr. Michael Gibson, Crystal McFadden, and Marla McKenzie for their continued support and expertise throughout this research project.